Hello, everybody. We're going to keep this respiration train going, and today's lecture is special because we will be pivoting into aerobic respiration, starting with the citric acid cycle. Our learning goal today will be illustrating the chemical logic and transformations that acetyl-CoA experiences throughout the citric acid cycle. We are going to be again introducing the mechanisms required to fully dismantle this molecule into energy. Our pre-class problem is actually a commercial product that you may have seen advertisements for or have seen on the shelves, and it is called 5-Hour Energy. It is an energy supplement shot, basically, that claims to provide a so-called crashless boost in energy by hyperdosing you on several water-soluble vitamins and other organic compounds. And this is a screenshot of its supplemental facts. We have 150% of our daily niacin intake, whopping 2,000% of our B6 intake, folic acid, a normal amount of that, over 8,000% of our daily B12 intake, and then this so-called energy blend consisting of taurine, glucuronic acid, malic acid, some amino acids, caffeine, whatever that is, so on and so forth. So we have spent the past several few lectures talking about how the cell produces ATP, okay? Now, we've also discussed how the cell upregulates or downregulates the catabolic machinery needed to do so. So my question to you is, which of the following active ingredients that I've listed off are responsible for stimulating which reactions in respiration? We left off our discussion with pyruvate processing through the hypoxic responses, such as fermentation and the Cori cycle. The flip side to all of that is the aerobic pathway that I've been hyping up. This aerobic fate of pyruvate is how we produce the bulk of glucose-derived ATP. Our goal over here is to prep the pyruvate molecule to enter the aerobic respiration pathways by oxidatively decarboxylating it. We will start with pyruvic acid and see this COO- highlighted in red. We will be removing that and oxidizing the pyruvate molecule through the enzyme pyruvate dehydrogenase complex. And when we do so, we will extract out NADH, which is again money in the bank. We will be eliminating that CO2, which is a great leaving group. And that will facilitate the inclusion of coenzyme A, which when we merge with pyruvate will obtain acetyl-CoA. That's one of our special energy currencies of the cell. It's a 2-carbon carrier, shown over there. This step is sometimes called the conversion step because what we are effectively doing is connecting anaerobic glycolysis to the aerobic citric acid cycle. And what is the chemical logic here? Well, we're making this entire carboxyl moiety in red a good enough leaving group to allow for coenzyme A to replace it. And it will do this through a very intricate mechanism. This mechanism is also very thermodynamically favorable. Its delta G is negative 33.4 kilojoules per mole. So it is favorable and hence irreversible and tightly regulated. The entities that allosterically activate pyruvate dehydrogenase complex include ADP, NAD+, calcium 2+, and coenzyme A. Think about the rationale here. ADP, NAD+, coenzyme A all mean we need more energy. So that stands to reason. Calcium gets released during active muscle contractions, which is also a sign that we have a high energy bill we need to play. The downregulators for pyruvate decarboxylase dehydrogenase is ATP, NADH, fatty acids, and acetyl-CoA. When these entities are in abundance, that means the energy demands of the cell is low and we have plenty of resources. No need to do all this catabolism. The pyruvate dehydrogenase complex is, well, a very complex series of 3 different enzymes, 5 different cofactors, all working together as a single protein that exists between the outer and inner membranes of the mitochondrion. The 3 enzymes involved are called shorthand E1, E2, and E3. And this is where things will get a little bit more messy. The E1 enzyme is called pyruvate dehydrogenase. And for every pyruvate dehydrogenase complex, there are 22 copies of this enzyme within its quaternary structure. Okay? E2, which is dihydrolipoyl transacetylase, that has 60 equivalents of that polypeptide baked in to this single protein. E3, dihydrolipoyl dehydrogenase, that's a measly 6 copies within the quaternary structure. Take a look at how complex the enzymes organize into the 3 domains. We have these E2s shown as these blue spheres. E3 is connected with those red versions, and E1 is shown there in yellow. The benefit of this very intricate arrangement of many different active sites is called channeling. One enzyme begins where another enzyme ends. So it's kind of like an assembly line. We can carry pyruvate from E1 to E2 to E3. And the benefit of that is we are reducing entropy. We are making the random path our substrate needs to navigate from A to B to C much more constricted and therefore entropically feasible. We will now examine the mechanism of every step. Again, I will not ask you to draw me the entire mechanism of any reaction from memory. On the test, I may simply ask you what the role of a cofactor is, or to use your intuition to fill in a small blank spot. As always, understand over memorize. Anyway, the core cofactor for E1 pyruvate decarboxylase is thiamine pyrophosphate. This is a derivative from vitamin B1. This vitamin is really special actually because it has a thiazole ring, this N double bond C connected to a single bond S. And this is really special because through resonance, we're able to obtain a carbanion on that middle carbon between the nitrogen and the sulfur. That carbanion is going to be a really good nucleophile, which makes sense because it's a lone pair that's dying to attack something that's a little bit electropositive. And what is the electropositive entity we are attacking, or aka the electrophile? It's going to be the carbon on pyruvate connected to that ketone. So we will be facilitating a backside attack over there via the carbanion. When we attack that C double bond O, we are again going to form a tetrahedral intermediate. See what a common player the tetrahedral intermediate is becoming in our mechanisms? Once we form that tetrahedral intermediate, the ensuing adduct is called the thiazolium adduct. And an image of its molecular structure is over here. Notice how it looks a little bit uncomfortable considering we have these explicit negative and positive charges on the same molecule. Nature abhors a charge. So the way we're going to fix that high energy state is to have the negative charges flow towards the positive charge. The purpose of vitamin B1 here is to impart a positive charge on the other side of the bond we're trying to break. So this O falls down. That will break the bond between this carbon and that carbon, and we will produce an alkene between the C1 of this portion and the carbon of our thiazolium. Because we have alternating lone pairs and double bonds, this high energy intermediate will also experience a degree of resonance. And one of those resonance forms obtains an orbital overlap that would be very prone to protonation, because we will promote an explicit negative charge on that carbon. By picking up a proton from a general acid residue, we will create something called the hydroxyethyl intermediates. There's an ethyl group, and there's an OH. This process is going to be the rate-limiting step of the entire pyruvate decarboxylase reaction. E2 and E3 of pyruvate dehydrogenase complex actually occur kind of together, and I promise it's not nearly as bad as it looks. The goal here in these two reactions is to remove this 2-carbon unit that we just superglued onto our thiamine. And to do that, we can remove this hydroxyethyl intermediate with another cofactor called lipoic acid. Lipoic acid is called a swinging arm coenzyme, and it actually has a lot of functional similarities to biotin. Within the anatomy of lipoic acid, it's essentially an octanoic acid molecule that contains a cyclic disulfide at the tail end. And usually this coenzyme is derivatized onto like a lysine residue, which is also very floppy within itself. And so what we're basically doing here is making a very long and floppy redox head that can grab onto a substrate in the active site and reel it on over to another site. And this is how it works. We will first, let's just recap the decarboxylation process. We have a carbanion form on our thiamine vitamin. The carbanion forms a covalent bond with our pyruvate molecule. once we do that, the negative charge on the carboxyl will swing down and break it off, yielding a hydroxyethyl intermediate. Okay? This hydroxyethyl intermediate is reduced. We can oxidize it by using this lipoic acid coenzyme. The electrons over here will transfer over to one of those sulfurs and the disulfide, and it's going to break open that ring and therefore oxidize our hydroxyethyl intermediate. Now we have a tetrahedral intermediate that we can then dismantle in the following manner. Immediately after that, a basic residue within the E1 site is going to deprotonate the alcohol of our attached hydroxyethyl intermediate, and that's going to rearrange the electrons to ultimately break the carbon-carbon bond between our intermediate and the coenzyme thiamine. The product of this reaction via the transacetylase domain is going to be acetyl-lipoamide, which I'm showing over here. It's like we're wiping off that 2-carbon hydroxyethyl onto the cyclic disulfide. Now that we've transferred our substrate from E1 onto E2, we will now be employing the transacetylase activity to basically exploit the fact that acetyl-lipoamide is also a good leaving group. This is a thioester, which are really easily broken. Acetyl-CoA, which possesses a thiol group, that's a nucleophile, will attack the carbonyl carbon in blue, and you're going to form a tetrahedral intermediate. The pi electrons will fall back down, and it's going to break one of these 2 carbon-sulfur bonds. The irreversible way will be breaking this one. That's going to liberate dihydrolipoamide, which is reduced and generate one equivalent of acetyl-CoA. This is what we need to proceed with the citric acid cycle. However, we're not done yet. We have to regenerate our enzyme to be in its original state so we can repeat this process with another additional pyruvate molecule. We need to reoxidize our dihydrolipoamide back into lipoic acid. That is where E3 comes into play. We will be using one equivalent of FAD, and we will react it with both of these thiols in the dihydrolipoamide molecule. Oxidizing 2 sulfhydryls together will give you a disulfide, shown here. And we will transfer these hydrides from that position onto FADH2. And then this FADH2 will transfer hydrides again to an NAD+ molecule, hence generating NADH. So this is really nice because we not only regenerate our pyruvate dehydrogenase enzyme, but we get a little bit of NADH out of the process. And remember, that is money in the bank. Now, why did we go through all of that effort to turn pyruvate into acetyl-CoA? Well, the short answer is that acetyl-CoA is what enters the citric acid cycle, which will fully strip the electrons from acetyl-CoA into reducing equivalents, such as NADH or FADH2. And shown in this figure, we have an overview of that process, where we combine a 2-carbon acetyl-CoA unit with a 4-carbon oxaloacetate, and the product of that is the titular 6-carbon citrate molecule of the citric acid cycle. And what we do from here on out in the following steps is employ a series of 8 different enzymes that will rearrange the functional groups on this 6-carbon backbone so we can systematically expose carbons for oxidation reactions. And every oxidation event, you can tell, is going to occur when catalyzed by something dehydrogenase. Isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, malate dehydrogenase. And what do all of these generate? NADH, NADH, NADH. Succinic dehydrogenase is a bit of an exception. It's still a redox enzyme, but it will be generating FADH2 instead, which has analogous activity. We'll get into the weeds soon enough, but first what I want to emphasize, just the big picture, citric acid cycle, it's the first part of aerobic respiration. It happens after glycolysis and after this intermembrane step. Once we convert pyruvate into acetyl-CoA, we will shuttle that intermediate into the matrix of the mitochondria, which is the innermost portion of this organelle. The first goal of the citric acid cycle is to fully oxidize acetyl-CoA into 2 carbon dioxide molecules, and it will do so again in 8 enzyme catalyzed steps. Equally important, the process of oxidizing acetyl-CoA will make 3 NADH molecules and 1 FADH, shown over here. What do I say? Think of these as money in the bank. The body has a way of converting these into a boatload of ATP in the final step of respiration, which is called oxidative phosphorylation. Now, another perk of the citric acid cycle is that we continuously make several useful intermediates. The citric acid cycle is leaky. We can bleed off citrate and make fatty acids, succinate and make some amino acids, oxaloacetate and make sugar, and we can use all of these as feedstocks for other metabolic streams. Let's first take a brief tour around the citric acid cycle before we discuss the mechanism of each step in detail. The first reaction of the citric acid cycle is also the last reaction. That's because it's a cycle, and hopefully by the end of this slide, it'll make sense as to why that is. The first enzyme in this process, citrate synthase, is going to catalyze the transfer of this red highlighted portion of acetyl-CoA onto a molecule of oxaloacetate. Oxaloacetate is a 4-carbon organic acid, and acetyl-CoA is a 2-carbon carrier. This is going to yield one equivalent of the 6-carbon tricarboxylic acid citrates, and it's going to recycle a coenzyme A for further cellular use. Now you may be wondering, oh my God, why are we about to go on this wicked journey? This seems to be unnecessarily complicated. If the goal is to get energy out of acetyl-CoA, why doesn't the cell just do that? Why are we sending it to this citric acid cycle? Well, it's actually not unnecessarily complicated. The energy remaining in acetyl-CoA is stored in a very tiny nut to crack, and it's easier to crack open a larger nut. I want you to think about sugar cookies, all right? If you've ever made sugar cookies before, you make the dough, you roll it out into a sheet, and then you take your cookie cutter, and then you punch out holes. And what happens when you're done making as many holes as possible? You're going to be left with a bunch of trimmings, but you don't want to throw that away. There's still a lot of cookie left over there. So what you do is you ball it up and maybe glom it on to a bigger ball of cookie dough, and we'll roll that one out again. So you have a fresh sheet with the old mixed in with the new, and then you can get more cookies out of it. No waste. That is what citrate synthase is doing. We're taking the trimmings of glycolysis, the acetyl-CoA fragments, and we're sticking them into a ball of cookie dough that the cell can more easily work with. We're going to systematically rearrange that acetyl fragment within the citrate scaffold. And these multiple rearrangements are necessary to thoroughly expose all of the bonds we intend to oxidize, and that will let us maximally harvest energy from this molecule. It's really quite ingenious. So we have our 6-carbon citrate molecule. 2 of those carbons came from acetyl-CoA. And now we're going to send it down the assembly line and have it be systematically oxidized by the citric acid cycle machinery. Well, there's one problem. Citrate is symmetric, and enzymes don't like working with symmetric molecules. It's hard to bind them in a specific manner, which means you can get scrambling, racemization, and all sorts of unpleasant modifications that create a lot of disorder in the substrate. So we're going to fix that with the next enzyme, aconitase. Aconitase is going to convert citrate into cis-aconitate and then isocitrate, which is an asymmetric version of our reaction namesake. It's an asymmetric citrate molecule. And now that isocitrate is asymmetric, the rest of the enzymes are going to have an easier time binding it and reacting with it in a way that will give us energy. And that's where the next enzyme comes in, isocitrate dehydrogenase. And remember, dehydrogenases are going to foster redox reactions. This will be the citric acid cycle's first instance of oxidative decarboxylation. We are going to remove one of the carbons in our 6-carbon isocitrate molecule, giving us a 5-carbon alpha-ketoglutarate. And in that process, we will be generating one equivalent of NADH, which we can use to make lots of ATP in the next step of aerobic respiration. Nice. So we put 2 carbons in, and we've already taken 1 carbon out. Pretty cool. The next enzyme, alpha-ketoglutarate dehydrogenase complex, is going to do something similar. It's another instance of oxidative decarboxylation. We're going to remove our second equivalent of carbon dioxide. And in the process, that's going to help us drive in another oxidation, meaning we get another NADH out of this reaction. This is going to leave us with the substrate succinyl-CoA, which is a 4-carbon compound. And hey, we started with 4 carbons back up here with oxaloacetate. We have removed 2 carbon equivalents halfway through the cycle. Now, what the other half of the cycle is going to be devoted to is rearranging the succinyl-CoA intermediate until we get back to oxaloacetate, which can feed back in to the first and therefore also last reactions of the citric acid cycle. The enzyme that acts upon succinyl-CoA is succinyl-CoA synthetase. And this enzyme is very nifty because it actually is going to do one instance of substrate level phosphorylation. And you may be thinking, hey, that's glycolysis's job, and that's true. But we have this very, very high energy coenzyme A group attached. And the mitochondrion is going to exploit that fact and use its hydrolysis to drive the formation of GTP or ATP, cellular energy. So that's a nice bonus that the citric acid cycle provides. A little bit more energy, right? So once we remove that CoA, we are left with the molecule succinate, and we are going to have another dehydrogenase act upon that intermediate, and it's going to oxidize succinates into fumarates. And if we're oxidizing succinate, we're reducing an energy carrier molecule. And the one that we are energizing or reducing is FADH2. This is like NADH in the sense that we can convert this into ATP currency. And you're going to again see that happen when we get to the electron transfer chain, which is coming next. And if you're wondering why it makes FADH2 and not NADH, don't worry about it for now. We'll blow up that bridge when we get there. But back into the cycle proper, we're now going to see fumarate be acted upon the enzyme fumarase, which is going to take this double bond that we formed in the previous step, and we're going to hydrate it into an alcohol. And again, everything here has a reason. Why are we just throwing in an alcohol? It's not for fun. The reason is alcohols are much easier to oxidize than alkenes, at least from the perspective of biochemistry. And so with our next step, malate dehydrogenase, hey, a dehydrogenase, we're going to oxidize that alcohol into a ketone, which is going to generate our third and final NADH equivalent. And in the process, it's going to convert malates into oxaloacetate, which is where we initially began. And this process can continue again and again, combining with another acetyl-CoA, and it's a really smoothly running, well-oiled engine. If this feels overwhelming, that's because it is. No one's going to grasp this all on the first go. So I encourage you to pause and rewind, and even then, if you're still a little shaky, don't worry about it, because we're going to go through these steps in detail, and I think the combination of the forest level perspective and the tree level perspective is going to slowly lay the foundations for understanding this entire complicated process. And I think in time you'll really appreciate the beauty of it. And now we dive into the individual enzymes. Step 1, citrate synthase. We take our oxaloacetate, a 4-carbon organic acid, and we combine it with our 2-carbon acetyl-CoA, and bam, we get citrate. The chemical logic of this step is that we employ thioester hydrolysis to drive condensation of the acetyl fragment onto that carbon of oxaloacetate. Not an easy step. Yet, it has a highly negative delta G of negative 32.2 kilojoules per mole, which means we have a thermodynamically favorable and hence irreversible transformation, which is of course regulated allosterically. ATP and NADH always indicates that we are in a state of energy abundance. Deactivate the enzyme, slow down the citric acid cycle. Additionally, acetyl-CoA, which is a product of a later stage in the cycle, will also shut down citrate synthase. That's an example of feedback inhibition. It's kind of like the opposite of feed-forward stimulation. We're going to have regulatory steps communicate with each other as well in the citric acid cycle. Of course, for our activators, ADP, that's going to speed up the enzyme because it means we're not making enough energy. A cool fact about the citrate synthase anatomy is that substrate binding is sequential. The binding of oxaloacetate is actually always going to form first. And in doing so, it is going to rearrange the fold of the protein to actually create the acetyl-CoA binding pocket, which is going to come in next. This is actually highly ingenious if you think about it, because if we bind acetyl-CoA first, there's a risk of it reacting with water and turning it into this coenzyme A alcohol instead. And that would be an absolute waste. If we ensure the proper substrate isn't present first, that's going to ensure we have the right nucleophile participating in the mechanism. The mechanism for citrate synthase is as follows. We have an aspartate serving as a general base. It is going to deprotonate a methyl group of all things, and that is going to upgrade this CC bond into a double bond. In doing so, we are increasing the bond order of this carbonyl carbon. So what do we have to do to make it happy? Break a pi bonds. We're not going to break the bonds that we just formed. How about we break the bonds between the C and the O? Because that oxygen can certainly tolerate a transient negative charge. Even more so, it is going to be stabilized by a protonated histone on the other side. This intermediate is called enoyl-CoA, and it is very reactive. And this highly unstable intermediate allows us to do a really challenging reaction next, an aldol condensation. Electron density swings back down, okay? And then we're going to break the pi bond we just formed between both of these carbons. That electrical density is then going to migrate to this pretty electropositive carbon between that double bond O and that COO minus. Two considerably powerful electron withdrawing groups. This is what drives the formation of a new carbon-carbon bond between acetyl-CoA, a methyl group, and oxaloacetate. This product is called acetyl-CoA, and this easily hydrolyzes in the presence of water to make citrates. The next enzyme in this cycle is aconitase, and in my opinion, its mechanism is a little hard to visualize, but we're going to do our best. Now, its job is to take the citrate molecule that we made in the previous step and dehydrate it. We are going to remove the hydroxyl group, shown in red over there, as a water molecule by protonating it. This is going to form the intermediate cis-aconitase, which is going to have a newly formed double bond right over there between the C3 and C4 carbons. And then we are going to take the water molecule that we just formed and slap it back in. We're going to rehydrate that newly formed double bond. But we're not going to rehydrate at the C3 position. Instead, we're going to rehydrate the C4 shown over here. And so what do we get out of that? Isocitrate, which is notably asymmetric. That is crucially important because citrate is a symmetric molecule. Breaking citrate's symmetry by moving its central alcohol down a position is one of the key bouts of chemical logic specific to aconitase. That's because symmetric substrates are more at risk of getting racemized by downstream enzymes, and many enzymes are regiospecific and enantiospecific. So ultimately what we're doing is reducing reaction entropy. So reducing entropy for later regiospecific chemistry. Furthermore, the alcohol in citrate is tertiary, and in isocitrate, it's secondary. Making it a secondary alcohol is going to generate favorable electronics for a subsequent oxidative decarboxylation reaction. And that will be more apparent when we look at the next mechanism, which features this reaction. And as challenging as this mechanism is to visualize, it's actually quite short, all right? The first thing aconitase will do is dock our citrate molecule onto this iron sulfur cubane, which is shown over here. Sulfur cubane. All right, it's pretty cool. It consists of 4 irons and sulfurs, respectively, that alternate in a literal cube-like fashion. So that iron-sulfur cluster is going to grab on at 2 different points, shown over here, there, and there. And once the substrate is in place, a basic residue B, usually it's a deprotonated serine, will attack this specific hydrogen atom on C4. So we'll call this Cr-, which will feature a negatively charged alkoxide, and it's going to attack like so. And really quickly, just to help keep you oriented, to help you find where C4 is, I'm going to highlight C1, C2, C3, and then C4. Over there. Anyway, back to the mechanism. Our deprotonated serine is going to do general base catalysis. It's going to attack that hydride, and that's going to cause those electrons to move over there and form a double bond between C4 and C3. And oh look, we just created a double bond. That means something needs to go on C3. Otherwise, it's going to have 5 bonds to it. And looking at the groups bounded to C3, only one of them is connected to an already polarized entity, and that's going to be this CO bond over here. That's the one that we are going to cut. The other options are carbon-carbon bonds, and those are going to be pretty stable and boring. So that hydroxyl group, the one attached to the cubane, is going to be our leaving group. Now, normally, hydroxyl groups are not good leaving groups. But there's going to be a nearby protonated histidine, and that's going to serve as a general acid. It is going to donate a proton to that hydroxyl, and it's going to turn it into an oxonium. And that resulting oxonium is going to be a great leaving group. It's going to hop right off, and that cubane will keep it in place for a sec because things are about to get spicy. Once we cut that bond between C and OH, well, really the oxonium, so it's going to liberate water, that means the citrate molecule, well, now it's cis-aconitate, is only attached to our iron sulfur cluster at one point, which is over here. That's the last remaining connection. So that molecule, that cis-aconitate intermediate, is free to swivel 180 degrees. It's going to rotate like so, turn all the way around, and this is where things get kind of messy, so I'll try to clarify up over here. We will have our iron bound to the water, and that is going to now be next to C4. The C4 is going to rotate above the C3. It's going to be double bound to C3, and nearby to the C3, is going to be our general base, which is going to be a protonated serine, but we'll just call it HB for now. We will now do the reverse of the first step of this enzyme mechanism. That histidine that we just deprotonated is going to want to get its protons back. It's going to attack that proton and activate that water that just left and turn it into a nucleophilic hydroxide. That will attack the C4 position, and that's going to cause this double bond to attack the proton that serine just plucked off of the C4 position. And when all is said and done, you're going to end up with isocitrate. What we literally just did was swap the positions of the hydroxyl and the H, highlighted in red and blue, respectively. So I hope you agree that the mechanism isn't terribly difficult to understand, it's just hard to draw on a crammed 2D plane. Now, there's something else important that I want you to know about aconitase. It's one of several moonlighting proteins in biology, and a moonlighting protein is a protein that has more than one role. Yes, aconitase's job is to be the second step of the citric acid cycle, But this iron-sulfur cofactor is very, very special. It actually serves as a cellular sensor for oxidative stress. So aconitase moonlights as an ROS sensor and CAC enzyme. Now, how does it do that? When the mitochondria get a little too overactive, they can generate reactive oxygen species like hydrogen peroxide or the superoxide radical. Or when the mitochondria is under a lot of stress, it can generate these radicals. And radicals are extremely toxic to cells. These reactive oxygen species, incidentally, are also good iron-sulfur cofactor destroyers, which means if a mitochondrion starts to go haywire, the reactive oxygen species they generate will shut everything down. It will cripple aconitase and put the brakes on the citric acid cycle hard, therefore stopping the production of more reactive oxygen species. So therefore, we say that aconitase is a moonlighter of radical oxygen species sensing and doing the citric acid cycle. So high reactive oxygen species is going to ultimately dismantle the iron sulfur cluster, which shuts down the citric acid cycle. Step 3 is catalyzed by isocitrate dehydrogenase. Our goal here is to oxidize our 6-carbon isocitrate into a 5-carbon alpha ketoglutarate. This is an important step because, like pyruvate dehydrogenase, it catalyzes an oxidative decarboxylation reaction. This is the first CO2 releasing step within the citric acid cycle proper. And this is also going to generate our first equivalent of NADH. We are oxidizing our substrate. That means we need to be reducing something else. And in this case, it's going to be an NAD plus molecule. So remember how in aconitase, the previous enzyme, we were relocating that hydroxyl group from C3, which used to be there, and we moved it to there in the C4 position? Well, what we effectively did was convert our tertiary alcohol into a secondary alcohol. Now, that was important because now we can oxidize it. We can't oxidize tertiary alcohols. And this oxidation of the highlighted alcohol is what ultimately gives us energy. Even better, oxidizing it into a carbonyl through the following transformation, see that over there, is going to convert isocitrate into oxalosuccinate, an intermediate, which is a beta keto acid. Converting the alcohol into a ketone also changes it from a weak electron donating group to a rather strong electron withdrawing group. And this is why beta keto acids are going to be prone to decarboxylation reactions. To clarify what I mean by that, the electron withdrawing effect is going to sort of act like a vacuum that's going to make the bond between this carbon and the carbon of this red highlighted carboxyl area very weak and very unstable. And when you throw in this manganese cofactor from the enzyme, Oh boy, it's like we crank up that electron vacuum effect all the way to 10. So follow those arrows. The electrons are going to flow from that carbon-carbon bond that's going to break off the carboxyl as a CO2. And those electrons are going to swing up onto that newly formed ketone as a CO minus, which gets stabilized by our manganese cofactor over there. Now, the intermediate that we get after that is an enolate, because we have a double bond next to a CO minus, and it is to a degree stabilized by that manganese cofactor, but it's not that stable. So it's going to rearrange back into a carbonyl that is now in the alpha position to the nearest carboxylic acid. Hence, alpha ketoglutarate. It was in the beta position of the COO group that we took off, but it is in the alpha position with respect to this carboxyl group. And I know I've said this before, but I'll say it again, that decarboxylations have very, very negative delta Gs because they are very entropically favored. Entropically favored. So this is not only a key regulatory step for the citric acid cycle, but it's also the rate limiting step of the citric acid cycle. It's its pacemaker. The key downregulators, of course, is ATP, and our stimulators will be ADP and calcium, 2+. So we're going to pause for now. We'll cover the rest of the steps in the citric acid cycle in the next video. But for now, I just want to leave you with these two bonus visuals. I know out of all the enzymes that we've covered so far, usually the one that students struggle with is the mechanism for pyruvate dehydrogenase complex. And so that's why I provided these two graphics that ultimately just show the same reaction in two different ways. And my recommendation is if you're struggling with following along mechanisms, is just look and compare as many different representations as you can. The chemistry in these two graphics are identical. It's just different ways of visualizing it. So try to follow through the arrows slowly in one version, and if that doesn't help, try the other, or if not, you can always come find me or we can talk about it more in class. But usually that's the trick. When one representation doesn't stick, just look at it from another perspective. All right, great work, and I'll see you in class.